Microorganisms show response to nutrient limitation and also to variation in the nutrient level. So, understanding of the environmental changes are very important, which help us to know about the ecological distribution of microorganisms, and it also helps in the control of microorganisms. Also, the ability of some microorganisms to adapt to extreme and inhospitable environment is truly remarkable, and this property of the microorganisms have been exploited for a number of applications in different fields. There are various environmental factors that determine the microbial growth, and it includes solute concentration of the environment. Changes in osmotic pressure will affect the growth of microorganism. It is also having a plasma membrane, which is a semi-permeable membrane. Concentration of nutrient outside the membrane and inside the cell will result into difference in osmotic pressure. Microorganisms are found in both hypotonic and hypertonic environment. When a microorganism is placed in a hypotonic environment, water leaves the cells into the environment, and the cell will get dehydrated and it will die. The net flow of water will be from inside to out to balance the osmotic pressure. So, in hypertonic environment, when the water leaves the cells, this will lead to the shrinkage of plasma membrane, and it is termed as plasmolysis, which subsequently damages the plasma membrane. On the other hand, when the microorganisms are placed in a hypotonic environment, water will enter the cells and it causes it to burst. This is called plasmoptosis. This image shows the effect of cells when they are placed in different conditions. You can see that in the hypertonic condition, plasmolysis takes place, and in the hypotonic condition, plasmoptosis takes place. That is, the water will enter and cause the cell to burst, whereas in isotonic condition, the solution contains equal concentration of impermeable solutes on either side of the membrane, and so the cell neither swells nor shrinks and remains flaccid. So now let's see how the microorganisms respond to such changes of osmotic pressure. When placed in hypotonic environment, the bursting is prevented by various mechanisms adopted by the microorganism to reduce the osmotic concentration within the bacterial cell. For this, some bacteria have mechanosensitive channels to escape the adverse effect of the hypotonic environment. When a bacteria is placed in a hypotonic environment, its plasma membrane stretches due to cellular swelling. This stretching opens the mechanosensitive channel and allows the solute to leave the cell, thus reducing the osmotic concentration within the cell and protecting the cell from bursting. And the second method is by the use of convertible vacuole, which acts as a pumping device to take an extra solute for balancing the osmotic pressure. Now, when the microorganisms are placed in hypertonic environment, it will result in shrinkage of plasma membrane or plasmolysis. In order to avoid that, many microorganisms make use of compatible solutes which help to keep the osmotic concentration of their protoplasm slightly above that of their surrounding. Compatible solutes are solutes that are compatible with the metabolism and growth of a microorganism, even if they are present in high concentration within the cell. That is, their high concentration will not affect the growth of the cell. Compatible solutes used by microorganisms include choline, betaine, proline, glutamic acid, sucrose, glycerol. Sometimes arbitol, sorbitol and mannitol are also being used. Organisms which require high salt concentration for optimum growth is referred to as halophiles. They almost require sodium chloride or other salts at a concentration above about 0.2 molar. Extreme halophile require high levels of salt ranging between 2 molar to 6.2 molar. Some microorganisms such as Halobacterium salinarium use potassium ions to raise their osmotic concentration. Organisms which can tolerate a wide range of osmotic concentration are referred to as osmotolerant organisms. For example, Staphylococcus aureus can tolerate wide range of salt concentration. They are also termed as halotolerant. Osmotolerant can also be defined in a different way based on the degree of water availability. In an area with low water activity, some organisms can maintain a high internal solute concentration to retain water, and such organisms are also termed as osmotolerant. Examples of osmotolerant include Aeromonas staphylococcus species, and example of halophile is Halobacterium halobium, Solanarium. The next factor that determines the microbial growth is pH. pH refers to the negative logarithm of hydrogen ion concentration. Microbial growth is strongly affected by the pH of the medium. Drastic variation in cytoplasmic pH disrupt the plasma membrane or inhibit the activity of enzymes and membrane transport proteins of the microorganisms. As hydrogen ion concentration increases, acidity increases, so the pH drops. As hydrogen ion concentration decreases, basicity increases and the pH goes up. Here you can see the classification of bacteria according to pH. And in this classification, 5 groups have been shown. And you can see here the extreme acidophiles having the pH range of 0 to 2. Acidophiles having the range of 2 to 5. And the neutrophils between 5 to 8. Alkalophiles between 9 to 12. And the extreme alkalophiles above 12, that is 12 to 14. So broadly, based on pH, the microorganism can be classified into acidophiles, alkalophiles and neutrophiles. Acidophiles have a growth range between the pH 0 and 5.5. And the examples include Ferroplasma, Thiobacillus thiooxidans, Sulfolobus, Acidocaldarius. These organisms require very low pH, that is high acidity. And the next one is the alkalophiles or the basophiles, which is having a growth range of pH between 7.5 to 14. And the examples include Thermococcus alkaliphyllus. And the third group, the neutrophils, have an optimum pH range between 5.5 to 8. And the examples include Staphylococcus aureus, E. coli, Pseudomonas, Lactobacillus, etc. Most of the bacteria are neutrophils. They maintain a neutral cytoplasmic pH by various mechanisms because usually they cannot tolerate pH values much below 4 to 5 range. So the microorganism must also adapt to change in environmental pH in order to survive. The plasma membrane may be relatively impermeable to protons. Neutrophils also exchange protons for potassium ions using an antiport transport system. In bacteria, small variation in external pH is corrected by sodium proton or potassium proton antiport systems. If the external pH drops below 5.5 to 6, organisms such as E. coli and Salmonella typhimurium synthesize a new array of proteins as a result of high ATPase activity in order to pump protons out of the cell. This is referred to as their acidic tolerance response. If the external pH further decreases to 4.5 or lower, the cell synthesizes chaperones such as acid shock proteins and heat shock proteins, which prevent acid denaturation of the proteins and aid in refolding of denatured proteins. Also, the microorganisms frequently change the pH of their own habitat by producing acidic or basic metabolic waste products. Internal buffering also helps to maintain a stable internal pH. Can control large changes in pH by adding phosphates. The next factor that affects the microbial growth is temperature. Temperature is the most important factor that determines the rate of growth, multiplication, survival, and death of all living organisms. Microorganisms are usually unicellular and their temperature varies with that of the external environment. At very high temperatures, the enzymes, carriers, and other proteins of the microbes get denatured. Microbial membrane also gets disrupted as the lipid bilayer melts. Hence, at high temperature, both cell function and cell structure are affected. At very low temperature, the cell membrane solidifies and the enzymes doesn't work rapidly, thus affecting the cell function. The temperature range at which an organism grows most well is referred to as the optimum temperature. Microbial growth decreases above and below the optimum temperature and is referred to as minimum growth temperature and maximum growth temperature. The lowest temperature at which organisms grow is the minimum growth temperature. The temperature at which the most rapid rate of multiplication occurs is termed as the optimum growth temperature. The highest temperature at which the growth occurs is termed as the maximum growth temperature. And a temperature only slightly above this point frequently kills the microorganisms by inactivating the critical enzymes. Based on their tolerance to temperature, microorganisms can be categorized as psychrophiles, psychotrophs, mesophiles, thermophiles, and hyperthermophiles. Psychrophiles grow well at 0 degrees Celsius and their optimum growth is at 15 degrees Celsius or lower. They are cold-loving microbes and they cannot grow above 20 degrees Celsius. Psychotrophs or facultative psychrophiles can grow at 0 to 7 degrees Celsius and the optimum between 20 to 30 degrees Celsius and the maximum around 35 degrees Celsius. Mesophiles, they have an optimum temperature around 20 to 40 degrees Celsius. They are moderate temperature loving microbes. Thermophiles have a growth range in 45 degrees Celsius and 80 degrees Celsius and their optimum is between 55 to 65 degrees Celsius. They are heat loving microbes. And in hyperthermophiles, the optimum growth is between 80 degrees Celsius and 100 degrees Celsius and it may grow to temperature of 120 degrees Celsius. The term psychrophile was first used by S. Kingdom and Nelson. They are extremophilic organisms that are capable of growth and reproduction in cold temperatures. And the examples include Oscillatoria, Chlamydomonas, nivalis, Methanogenium. Psychotrophs are facultative psychrophiles. They are cold-tolerant bacteria. Psychotrophic bacteria and fungi are the principal cause of spoilage of refrigerated food and the examples include Pseudomonas, Aeromonas, Bacillus, Clostridium. Mesophiles. Most microorganisms, including all human pathogens, fall in this category and the examples include E. coli, Streptococcus pneumoniae, and they grow best in moderate temperature that is between 20 degrees Celsius to 45 degrees Celsius. Thermophiles is derived from the Greek word thermotita meaning heat and philia meaning love. They are heat-loving microorganisms. They grow at high temperature, hence they have more heat-stable enzymes and protein synthesis systems. The membrane lipids are also temperature stable. They are more saturated and more branched. The examples include Thermus aquaticus, Geogemma barrosi, Thermoplasma acidophyllum. Hyperthermophiles thrive in extremely hot environments. It was first discovered by Thomas D. Brock in 1965 in hot springs in Yellowstone National Park, Wyoming. Its cell membrane contains high level of saturated fatty acid to retain its shape at high temperature, and the examples include Sulfolobus, Methanococcus janashi, Thermatoga, Pyrococcus abscessi. The next factor affecting microbial growth is the oxygen concentration. Different microorganisms have different oxygen requirements. Based on the oxygen requirement, the microorganism can be classified as obligate aerobe, facultative anaerobe, aerobe-tolerant anaerobe, strict anaerobe, and microaerophiles. First, let's talk about aerobes. Organisms which require atmospheric oxygen for their growth. They are termed as aerobe. In that, the obligate aerobes cannot grow in the absence of oxygen. Here, oxygen acts as the terminal electron acceptor for the electron transport chain in aerobic respiration. An example of aerobic organism is algae. Next, let's talk about anaerobes. Anaerobes are organisms which do not require oxygen for their growth. Anaerobic bacteria usually generate energy through fermentation or other anaerobic respiration pathways. Anaerobes are of different kinds. First one is the facultative anaerobe. These organisms do not require oxygen for their growth, but they can grow better in the presence of oxygen. In the absence of oxygen, they produce energy by fermentation, whereas in the presence of oxygen, they produce energy by aerobic respiration. Example of this category is E. coli. The next group is the aerobe-tolerant anaerobes. They are anaerobes which do not require oxygen for growth, but the presence of oxygen do not affect its growth. They can tolerate oxygen even though they don't use it to produce energy. Example of this category is Enterococcus faecalis. The next group is the obligate or strict anaerobes. These organisms cannot tolerate oxygen at all. They die in the presence of oxygen. Example of this group is bacteroids, Fusobacterium, etc. The next one is the microaerophiles. They are organisms that require only low levels of oxygen for their survival. Normal atmospheric level of oxygen is harmful to them, but low concentration of oxygen is essential. That is, it requires less than 2 to 10% of oxygen. Example of organism that is coming under the microaerophile is the Campylobacter. And this image also shows the distribution of microorganisms in various tubes based on the concentration of oxygen. In the case of aerobic respiration, oxygen acts as the terminal electron acceptor of the electron transport chain. Oxygen accepts the electron and becomes reduced to form reduction products such as superoxide radical, hydrogen peroxide, and hydroxyl radical. These reduction products are highly toxic to the microbial cell. Hence, microbes making use of aerobic respiration have enzymes such as superoxide dismutase and catalase, which catalyzes the destruction of these harmful reduction products. And some use manganese ion to destroy this superoxide. The next factor affecting the microbial growth is pressure. Microbes obtain almost all their nutrients in solution from surrounding water. Most microorganisms living on land or on that water surface are subjected to only the normal atmospheric pressure of 1 atmosphere. Hence, they are never significantly affected by changes in pressure. But organisms living in deep sea are subjected to high pressure. Hence, these organisms are adapted to live under such high pressure. And such organisms can be classified as barrow-tolerant organism. They are organisms which are usually grown at normal atmospheric pressure, but they are able to tolerate high pressures also. And the other group is the barophiles or the pisophiles. They are organisms which can grow only at high pressure. They are unable to grow at lower or normal atmospheric pressure. And examples of barophiles include Halomonas, Salaria, Photobacterium, Shivanella, Cynophores. The next factor affecting the microbial growth is the radiation. Sunlight is a major source of radiation on the earth. Visible region of sunlight is the most important aspect of the environment, which is essential for life to be sustained on earth. Infrared is a major source of earth's heat, and ionizing rays can produce mutations which may result in death. The visible light is beneficial because it is the source of energy for photosynthesis. Microorganisms have pigments like chlorophyll, cytochrome, flavin, and can absorb light energy and get excited or activated, and they can act as photosensitizers. And these photosensitizers can transfer energy to oxygen and produce synclet oxygen, which is very harmful to microorganisms. Ionizing radiations have very short wavelength and high energy. They can cause atom to lose electrons or ionize. Ionizing radiation in low levels will cause mutation, which will gradually lead to death of the organism. High levels of ionizing radiation directly kill the microorganisms. Two major forms of ionizing radiation include X-rays and gamma rays. Ionizing radiation breaks down hydrogen bonds, oxidizes double bonds, destroys ring structures, and this will lead to the destruction of DNA. Non-ionizing radiations include UV radiation. The main mechanism of UV damage is by the formation of thymine dimers in DNA. That is, there is formation of covalent linkage between two adjacent thymine residues, and this dimer formation will inhibit the DNA replication. Exposure to near UV radiations also affect microorganisms. They cause tryptophan breakdown into toxic photoproducts which produce breaks in DNA strands.